Hey guys, welcome back to another YouTube video of my channel. Today we are going to discuss uh, chapter 2 from class 12th biology, that is sexual reproduction in flowering plants. Okay, so basically sexual reproduction means involvement of two parents. In flowering plants we'll see. So first of all, for the reproduction part we'll have to see what are the parts of a flower. So in a flower there are four different sets of whorls or floral members attached to the central axis called the thalamus. The central axis is called the thalamus. Outermost and the first accessory whorl is of sepals called the calyx. Second accessory whorl is of petals and is called the corolla. Petals, you know, the colorful part, and sepals is the green part, leaves and all that. Okay, next to corolla is the male reproductive whorl of stamen called the androecium, and next to it is female reproductive whorl, sorry, which is of carpel, is called gynoecium or pistil. Calyx calyx and corolla are the non-essential whorls, while androecium and gynoecium are the essential whorls because they are included in the sexual reproduction. Now I'll show you with the help of diagram. The outermost, first, this is the thalamus, sorry, this is the thalamus part. Okay, then when we move inward we see the sepals part, right? Then when we move next, we see these petals. Okay. Then when we move next, we see the male reproductive part, which includes the anther and the filament. We'll see in detail. This is the stamen part. And then when we move to the next, we see the pistil part. This is the pistil. It includes stigma, style, and ovary. I hope it is clear to you. Now, let's look at the function of the floral members. First, calyx. What, according to you, think calyx is of function? Let's discuss. Main function of calyx is to protect the flower in bud condition because this covers up the this part, it covers up the entire flower when it is in the budding stage. So though it is smaller than petals in size, but it performs protection when it is in the bud stage. When green, it shows it is photosynthetic in nature, and when colored, it attracts insect for pollination, right? I'll write here calyx includes the sepals so that there is no confusion, and corolla includes the petals, right? Okay. Corolla provides color and scent to attract pollinators. It is so easy. Sometimes its base bears some secretory glands and secretes nectar to provide smell to the flower. Obviously, another factor, but it is responsible for the same factor, that is pollination. Basic function is for the attraction, to attract the pollinators and to conduct the process of pollination. It also provides protection. It is a secondary function. Primary function is pollination. We have androecium. Androecium main function is production of microspore, that is pollen grains containing male gametes within the anther lobe. Right? It is the, you can say, the male reproductive organ. Right? Reproductive. Okay. And gynoecium. Gyno means female. So it is the female reproductive organ. Right? Its main function is the production of megaspores, that is eggs, fruits, and seeds. Remember, megaspores is in the females and microspores is in the males. You might get confused, but you need to remember this. It is a main thing. Now let's move on to the male reproductive unit. First we see stamen. Stamen, I'll show you roughly now. This is the stamen. This is the filamentous structure and this is the anther part, right? Male reproductive unit consists of the following two parts: long slender stalk filament and a bilobe terminal structure called the anther. I showed you this. I'll show you with the diagram also. Structure of anther. Structure of anther, it is composed of two anther sacs or lobes separated by a tissue called connective tissue. Anther is bilobe, and each lobe or sac consists of two theca separated by a septum. You can see here, this is the anther part, this is the filament part. When we have taken the transverse section like this, it is cut out in this plane, then you can see there are two lobes, right? And each lobe or sac contains two theca state, this and this and this. So it is a bilobe structure, right? Two lobes. One is this, one is this. In this diagram, you can see, oh, sorry. In this diagram, you can see one lobe is this one and one is this one. This is somewhat like this. Two lobes, right? And each lobe consists two thecas, right? I hope it is clear. Anther is a bilobe, and each lobe or sac consists of two theca separated by a septum. You can see a little bit of septum is visible, right? It is in then it is tetragonal, four-sided structure. You can see tetragonal, what I mean by this is somewhat like this, tetragonal. Okay, I know the drawing is not that good, but hope you understand. I've shown here also tetragonal in structure consisting of four microsporangia, two in each lobe. Okay, you can see here, if I show you, wait, now I'll show. Each theca we were calling, they are also known as microsporangia. These are called the microsporangia. Okay, microsporangia develops and get transformed into pollen sacs. They get mature, developed, and forms the pollen sacs. These pollen sacs then give rise to the pollen grains. These pollen grains are also known as the, also sometimes referred to as the microspores. Okay, each pollen sac gives rise to many of the pollen grains or the microspores. Now see, let's see how. Structure of the microsporangium, very important topic. Typical microsporangium may appear circular and is surrounded by four walls. Okay, there are four walls. I know it. Sometimes when I was a student, I used to get confused so much. Then I learned by this technique, epi and meta are friends. Epi and meta are friends. Okay, so epi, epi for epidermis, end means endothecium, me for middle layers, and te for tapetum. Now let's see. Epidermis is the outermost single layer of cells which is protective in nature. Endothecium, second layer, thick cells, helps in dehiscence. What is dehiscence? I am telling you. This give rise, earlier it is a bilobe structure when it is undeveloped, somewhat like this. Okay, so it is somewhat circular, then it gives a curvature type structure, and then it helps in the division. This division or separation, you can say, is the dehiscence. Also, it is protective in nature. Main function is of protection and dehiscence. First, epidermis is protective in nature. Next is middle layers. It is the third layer composed of 1 to 3 layers. Important point. It is the only layer which is composed of 1 to 3 layer of cells. It also helps in dehiscence and protective in nature. Majorly, these three layers are protective in nature, but tapetum is for the provision of nourishment. Important point. Nourishment. Right? Now let's see. It is the it is the fourth and innermost layer of cells with dense cytoplasm and many nuclei. It provides nourishment to the developing pollen grains. This is the important point for tapetum, that it provides nourishment to the developing pollen grains. The center of each microsporangium is filled with closely arranged similar cells called the sporogenous tissue. I'll show you this thing in the diagram. At maturity, pollen sac is formed by fusion of two microsporangium in each lobe. Okay, a mature anther dehisces by slits and liberate the pollen grain. Dehiscence, separation. Separation occurs to liberate the pollen grains, right? Now I'll show you diagram. This is the anther lobe. Okay, the outermost layer, epidermis. Then the endothecium, this is the endothecium. Okay, then this green part is the middle layers. Okay, then this yellow part is the tapetum, and inside tapetum is the sporogenous tissue. Okay, and all these four microsporangias are connected by a connective tissue within. Okay, now there it is a given on enlarged view. Okay, epidermis, endothecium, middle layers, microspore mother cells. What I told you microspore mother cells. Sorry, microspore mother cells. These cell only develop, mature, undergo dehiscence to form the pollen grains. And these cells, tapetum cells, they provide nourishment. Okay. Now, the formation of pollen grains from the pollen mother cells occurs through the process of microsporogenesis. Okay. Now, let's see the process. The process of formation of microspore from a pollen mother cell by meiosis. What is meiosis? Reductional division. Okay. It's called microsporogenesis. This is again transverse section of a mature anther with pollen grains. See, all the layers are there, and the sporogenous tissue is now lost. Dehiscence here occur, they get separated, and the pollen grain mature and come out. Okay, the pollen grain comes out. Okay, each cell of the sporogenous tissue. I've showed you sporogenous tissue inside the tapetum. They were situated in a microsporangium. What is a microsporangium? This entire is a single microsporangium. Each anther has four microsporangias. Okay, sporangia. Okay, remember this thing. Okay, each cell of sporogenous tissue in a microsporangium acts as a potential mother cell. Okay, each cell, there is a one microsporangium and there are millions of cells inside it, and each cell acts as a potential pollen mother cell. That is, it gives rise to a pollen grain, or you can say a microspore, or the microspore mother cell also we can call. PMC, that is the pollen mother cell, undergoes meiotic division. I told you reductional division. It uh, it keeps on dividing to form cluster of four cells called the microspore tetrad. Okay, first a single cell divides into two. These two form another two. Okay, these then combine together to form the microspore tetrad. Okay, on maturity, anther dehydrates and microspores separate from each other to form pollen grain. Now, when the anther gets mature and dehydrated, these earlier they were together. See, you can see if in this diagram you can see they are placed close to each other. When anther gets dehydrated, then these microspores separate from each other to form the pollen grains. With a two-layered wall, the outer hard exine and the inner intine. Okay, pollen grain. Now let's see what is a pollen grain, its structure, function, and everything. Generally, it is spherical in structure, possess two prominent wall layers, that is the outer exine and the inner intine. Okay, exine is the hard layer made up of sporopollenin, which is one of the most resistant organic material present in the nature. It can withstand. Why the exine is made so hard? You'll get to know later on. It consists of sporopollenin, which is the most resistant organic material. This is a very important point from the NEET point of view. Sometimes they make question of one or two questions from this topic itself about exine. That it is the most resistant organic material. It is made up of sporopollenin. Okay, it can withstand high temperature, strong acids, and alkalis. Even enzyme cannot degrade sporopollenin. That is why if we see the structure of enzyme, there are formed germ pores. Okay, somewhat like this. From these germ pores only, the pollen it comes out, and the further reproduction takes place. Okay, hence pollens are preserved as fossils. These are preserved as fossils. This is also an important point. Inner thin layer of intine is made up of cellulose and pectin. This is also asked sometimes in NEET, that the inner thin layer, it is made up of two elements, that is cellulose and pectin. Sometimes it is together referred to as cellulopectin, right? Exine has an aperture, right? See, I told you exine has an aperture where sporopollenin is absent. It is called the germ pores. It is also an important point. The newly differentiated pollen grain has a central nucleus and a dense cytoplasm. Pollen grain is the first cell of male gametophyte. I told you. These pollen grain are the first cell of male gametophyte. Okay, protoplast increases in size and mitotically divides into two unequal cells. Okay, these pollen grain, male gametophyte. Okay, the entire protoplast, this entire region, protoplast. Okay, it gets divided into two unequal cells. The bigger vegetative cell, this somewhat like this division occurs. This is the vegetative cell and this is the generative cell. Okay, rich in food reserve and large irregularly shaped nucleus. They have a single nucleus which is large and irregular in shape. Smaller spindle shaped generative, somewhat like this shaped. Okay, generative cell with dense cytoplasm, because it is smaller in size, so the cytoplasm is dense, it comes together. Okay, and a nucleus. This is a this is called the two cell stage. In majority of angiosperms, that is 60% angiosperms, pollens are released in this two cell stage. Okay, but in some rest 40%, the generative cells also undergo division. They form two male gametes, and thus pollen is said to be in three cell stage. Okay, now let's see. This is the microspore mother cell. It underwent meiosis. It formed the microspore tetrad. This. Okay. Then when the anther got anther mature and dehydrated. So these microspore got separated and released. Okay. Okay. This now these, when we see a single microspore, this is a single microspore. Okay, then it underwent growth and development, so it formed two vacuoles and a nucleus. Then it again went undergo maturity, it forms an asymmetric spindle. Then it forms the two walls, exine and intine. This is the outer exine, this is the intine, and a small spindle shaped, I told you, spindle shaped generative cell. It occurs, this is the two cell stage. Okay, then it again went mitosis. Okay, to form the larger some in 60% cell, it is the division is up to here only, but only in 40% cases this occurs. Okay, three celled pollen, two male gametes are formed because it undergoes mitosis. What is mitosis? Equational division. Okay, and vegetative cell is as it is, and only the generative cell undergoes mitosis to form three cell stage. I hope it is clear till here. Now only, now we will do importance of pollen grains. It contains male gametes for sexual reproduction. They are nutrient rich and thus are taken as food supplements. This is a very important point. It is nutrient rich and thus taken as food supplements. Pollens can be stored for years in liquid nitrogen at 196 degree, that is called the cryopreservation. The process of storing any egg, sperm, pollen grain or some other things, some other germ cells under liquid nitrogen at 196 degree minus 196 degree Celsius, it is called as the cryopreservation. This is also an important point. It can be asked to be used later in crop breeding programs, because sometimes we delay the process of reproduction, so it is stored. They may cause allergies and bronchial infections in some people, leading to asthma and bronchitis. This is also important. They are highly allergic in nature. Now, next topic, that is female reproductive unit, we'll discuss in next lecture. I hope you like the video. If you have any doubt, please drop down in the comment section. Thank you. If you like the video, please like, share and